All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
After being done with glycolysis in the Krebs cycle, we're left with 10 NADHs, and 2 FADH2s. And I told you that these are going to be used in the electron transport chain, and they're all sitting in the matrix of our mitochondria. And I said they're going to be used in the electron transport chain in order to actually generate ATP. So that's what I'm going to focus on in this video. The electron transport chain. And just so you know, a lot of this stuff is known, but some of the details are actually current areas of research. People have models and they're trying to substantiate the models, but things are happening at such a small scale here that people can just look at the evidence, some of it of which is indirect, and say this is probably what's happening. Most of this is very well established, but some of the exact mechanisms of, for example, how exactly some of the proteins work aren't completely understood. So I think it's very important for you to understand that this is kind of at the cutting edge. You're already there. So the basic idea here is that the NADHs, and that's where I'll focus, FADH2 is kind of the same idea, although its electrons are at a slightly lower energy state, so they won't produce quite as many ATPs. Each NADH, maybe I'll write it here, each NADH is going to be, as you'll see, indirectly responsible for the production of 3 ATPs, and each FADH2 in a very efficient cell, in both of these cases, will be indirectly responsible for the production of 2 ATPs. And the reason why this guy produces fewer ATPs is because the electrons that he has going into the electron transport chain are at a slightly lower energy level than the ones from NADH. So in general, I just said indirectly. How does this whole business work? Well, in general, NADH, when it gets oxidized, so NADH, remember, oxidation is the losing of electrons or the losing of hydrogens that happen to have electrons. We can write its half reaction like this. Its oxidation reaction like this. You'll have some NAD+, which you can then go and use back in the Krebs cycle and in glycolysis. You have some NAD+. You'll have a proton, right? A positive hydrogen ion is just a proton. And then you'll have 2 electrons. This is the oxidation of NADH. Oxidation of NADH. It's losing these 2 electrons. Oxidation is losing electrons. Oil rig. Oxidation is losing electrons, or you can imagine it's losing hydrogens from which it can hog electrons. Either one of those is the case. Now, this is really the first step of the electron transport chain. These electrons are transported out of the NADH. Now, the last step of the electron transport chain is you have 2 electrons, and you could view it as the same 2 electrons if you like. 2 electrons plus 2 hydrogen protons. And obviously, if you just add these 2 together, you're just going to have 2 hydrogen atoms, which is just a proton and electron, plus 1 oxygen atom. So I could say 1/2 of molecular oxygen. That's the same thing as saying 1 oxygen atom. And you're going to produce, if I have 1 oxygen and 2 complete hydrogens, I'm left with water. I'm left with water. And you could view this, we're adding electrons, or we're gaining electrons to oxygen. Oil rig. Reduction is gaining electrons. So this is the reduction of oxygen to water. This is the oxidation of NADH to NAD+. Now, these electrons that are popping out of, these electrons right here, that are popping out of this NADH. And when they're in NADH, they're at a very high energy state. And what happens over the course of the electron transport chain is that these electrons get transported to a series of, I guess you could call them transition molecules. But these transition molecules, as the electrons go from one to the other, they go into slightly lower energy states. And I won't even go into the details of these molecules. One is coenzyme Q, and cytochrome C. Cytochrome C, and then they eventually end up right here, and they're used to reduce your oxygen into water. Now, every time an electron goes from a higher energy state to a lower energy state, and that's what it's doing over the course of this electron transport chain, it's releasing energy. It's releasing energy. It's releasing energy. So energy is released when you go from a higher state to a lower state. When these electrons were in NADH, they were in a higher state than they are when they bond to coenzyme Q. So they release energy. Then they go to cytochrome C, release energy. Now that energy is used to pump protons across the cristae, across the inner membrane of the mitochondria. This is all very complicated sounding. And this is the cutting edge. This is the cutting edge. So it maybe should sound a little complicated. So let me draw a mitochondria. So let me draw a small mitochondria just so you know where we're operating. That's its outer membrane. And then its inner membrane, or its crista, would look like that. Maybe it looks just like that. And let me zoom in on the membrane. So let's say if I were to zoom in right there. So if I were to zoom that out, that box would look like this. You have your crista here, and I'm going to draw it thick. Right, so I'm zooming in. This green line right here, I'm going to draw it really thick. I'm going to color it in with green, just like that. And then you have your outer membrane. This outer membrane, I can do it up here, and I'll just color it in. You don't even have to see the outside of the outer membrane. Right here, this space right here, this is the outer compartment. And then we learned in the last video, this space right here is the matrix. This is the matrix. This is where our Krebs cycle occurred and where a lot of our NADH, or really all of our NADH, is sitting. So what happens is every time NADH gets oxidized to NAD+, or and the electrons keep transferring from one molecule to another, it's occurring in these big protein complexes. And I'm not even going to go into the details on this. So each of these protein complexes span, so let's say that's the protein complex where this first oxidation reaction is occurring and releasing energy. And then let's say there's another protein complex here where the second oxidation reaction is occurring and releasing energy. And these proteins are able to use that energy to essentially pump, it might all seem very complicated, to essentially pump hydrogens into the outer membrane. It actually pumps hydrogen protons, and let me be very clear, hydrogen protons into the outer membrane. And every one of these reactions pump out a certain number of hydrogen protons. So by the end of the electron transport chain, or if we just followed one set of electrons, by the time that they've gone from their high energy state in NADH to their lower energy state in water, by the time they've done that, they've supplied the energy to these protein complexes that span our cristae to pump hydrogen from the matrix into the outer membrane. So really the only byproduct of the oxidation of NADH into eventually water, or the oxidation of NADH and the reduction of oxygen into water, isn't ATPs yet. It's just this gradient where we have a lot higher hydrogen proton concentration in the outer compartment than we do in the matrix. Or you could say that the outer compartment becomes a lot more acidic. Remember, acidity is just hydrogen proton concentration, the concentration of hydrogen protons. So the byproduct of this, all of this energy is used to really just pump these protons into the outer membrane. So you have two things. The outer membrane becomes more acidic than the matrix inside. Maybe we could call that basic. And then obviously these are all positively charged particles. So there's actually an electric gradient or electric potential between the outer membrane and the inner membrane. This becomes slightly negative, that becomes slightly positive. These guys wouldn't naturally do this on their own. If this is already acidic and it's already positive, left to its own devices, these more protons wouldn't be entering. And the energy to do that is supplied by electrons going from high energy state in NADH to going to a lower energy state eventually on the oxygen in the water. That's what's happening. But essentially all that's happening is protons being pumped from the matrix into the outer compartment. Now, once that gradient forms, these guys want to get back in. These guys want to get back in to the matrix. And that is where the ATPs are formed. So there's a protein that also spans this. Let me draw. So remember, this is all this inner membrane right here. Let me just draw it a little bit bigger right here. So that's our inner membrane, our cristae right there. There's a special protein called, and I'll show you actually a better diagram of what it looks like in a second, called ATP synthase. ATP synthase. And what happens is, remember, because of the electron transport chain, we have all of these hydrogen ions up here. All of these protons, really. All they are is a proton that really want to get back into the matrix down here. They really want to get back down into the matrix, but they can't. This cristae is impermeable to them, so they have to find a special way to get through. They were able to go the reverse direction through these special protein complexes. Now they're going to go back into the matrix through ATP synthase. So they're going to go back, but something interesting happens. And this is really an area of current research where people are, they think they know how it works, but they're not sure, because you can't just take these proteins apart and operate them or watch them operate like you can a regular mechanical engine. These are ultra small, and they have to be in a living system, and they have to have the right conditions, and you can't even, it's hard to see hydrogen protons. These are ultra small things that are, pretty much you can't see them. But what happens, the current model is as these enter, as these go through my ATP synthase, there's actually an axle. So you can kind of view this as a housing, and then there's an axle. And this is all just a big protein. And there's an axle, and then there's another part of the synthase down here. So you can imagine, this is kind of mind-blowing that something this fancy is occurring on the membranes of pretty much all living systems cells. It's not just eukaryotes. Even prokaryotes do this. They don't do it in mitochondria. They do it in their cellular membrane. But it's a pretty neat thing. And what happens is, as these go through, you can kind of imagine as water flowing through a turbine, it mechanically causes this structure in the middle to spin. To actually spin. This is the current thinking. And this thing is all uneven. It's not like this nice tube, it'll look all crazy like that. And what happens is that an ADP molecule, let's say that this is the A part of the ADP, and then you have two phosphate groups. It'll attach to one part of this protein, and maybe a phosphate will just randomly attach to another part of this protein, just like that. So right now it's just ADP and a phosphate. But as this inner axle turns, because it's not a symmetrical tube, it has different things sticking out that has different amounts of atomic charge, and it's going to play with this outer housing right here. And so as this turns, the outer housing, because of just the proteins bumping against each other and electrical charge and whatever else, it's going to squeeze the ADP and the phosphate together to actually form ATP. And actually, the current thinking is it does it on 3 different sites simultaneously. So as this spins around, ADP and phosphate groups kind of show up on the on the inside of this housing. You could imagine like that. And I don't even know if it's on the inside, but they show up on the housing. And then as this thing spins around, it stretches and pulls on this outer part and pushes these two things together. So it's using the energy from this proton gradient to drive this axle. And because it's all strange, it does all these distortions on this outer part and actually pushes the two ATPs together. So when you start off with your 10 NADHs, it'll provide just enough energy to do just enough protons to put into the outer membrane that when they go back through our ATP synthase, you can almost view it our ATP synthase motor, just based on people's observations, they see that this will produce on a per NADH level roughly 3 ATPs. On a per FADH2 level, roughly 2 ATPs. And I've said multiple times in the videos that this is kind of an ideal. That a lot of times maybe you'll have some protons leak, so their energy can't be captured properly. Or maybe some of these electrons might somehow jump the gun or jump some steps, so some of the energy gets lost. So you don't always have a completely efficient system. And just so you believe that this is actually occurring on our membrane, there's actual visual depictions of these proteins. This is the actual protein structure of ATP synthase right here. That is actually ATP synthase. And as you can see, there's this piece right here that holds this part, that holds that part and that part. You can kind of imagine it relatively stationary. The hydrogen comes through here. The axle gets spun. And as the axle gets spun, ADP and phosphate groups that are lodged inside this F1 part of the protein get pushed together. You have to put energy into the reaction in order to make them stick together, but they get pushed together by the protein itself as this axle turns around. And this axle turns around from the energy of the hydrogen going. I don't even know what the mechanics would look like, but you could imagine, in my head, I imagine kind of, the simplest thing is a windmill, or not a windmill, is some type of water turbine, or maybe the simplest thing is, if you have something like that, I don't know if that's what the protein actually looks like. You have any kind of thing passing by, it's going to spin it. It's going to spin it like that. And you could be more creative if you want to change the angle of the spin and whatnot. And that's all, people are really still trying to understand this at a deeper and deeper level. But for your purposes, especially in an introductory biology level, you just have to realize that two things are happening in the electron transport chain. Electrons are moving from the NADHs and the FADH2s to eventually show up and reduce the oxygen. And as they do that, they're releasing energy as they go from one molecule to another. They're going to lower energy states. That energy is used to pump hydrogen protons into the outer compartment of the mitochondria. And then that gradient, those hydrogen protons want to get back into the matrix of the mitochondria. So as they go back in, that drives this ATP synthase engine, which actually produces the ATP. So just like we said in the past, when you have 10 of these, on average, let me say it this way, on average, each NADH is going to produce 3 ATPs. Not directly. It produces enough of a gradient of hydrogen protons to produce 3 ATPs in the ATP synthase. And each FADH2, on average, produces enough of a hydrogen gradient to produce 2 ATPs. So if we come in with 10 NADH, they're going to produce, in this ideal world, 30 ATP. And then our 2 FADHs, H2s, are going to produce 4 ATP. And then if you remember, from glycolysis, we had 2 net ATPs directly produced. And from the Krebs cycle, we had 2 ATPs directly produced. So then you have 4 from glycolysis and Krebs. And that gets us, once again, to our magic 38 ATP from one molecule of glucose. And now I think you have a pretty good grasp of cellular respiration. I made a slight error in the electron transport chain video, and I wanted to correct it in this one. And it's also an opportunity for me to include a little bit of terminology that I forgot to include in that video. So when I described the electron transport chain, we remember it's just you have some high energy electrons in NADH, and they get transferred from one molecule to another. And as they get transferred, they go into lower energy states, and they release energy. And then the final electron acceptor was oxygen. Oxygen got reduced right here. But if you look at both sides of this equation, the mistake was I need two hydrogens. If I have two hydrogens on the right-hand side in the water, I need two hydrogens on the left-hand side. So there should be a 2 right there. So that was what I would consider to be a minor mistake in the last video. But this also gives me a chance to introduce you to some more terminology. So this whole process, we know that this is called oxidation, right? When NADH loses a hydrogen, remember oxidation is losing formally electrons, but when it loses the hydrogens, it loses the opportunity to hog that hydrogen's electrons. So this whole process of the electron transport chain is one molecule after another getting oxidized until you have a final electron acceptor in water. So this is obviously you could call this oxidation, just very generally. And then the second part of the electron transport chain, or maybe we shouldn't even call this part of the electron transport chain, the process where the ATP is actually formed, the adding of a phosphate group to another molecule is called phosphorylation. Phosphorylation. So the whole process of creating ATP through the electron transport chain. Remember, the electron transport chain releases energy that creates this hydrogen gradient. It pumps the hydrogens into the outer compartment, and then that gradient, those hydrogens that want to get back into the matrix, essentially go back through this ATP synthase. This process of generating ATP this way is called oxidative phosphorylation. Oxidative phosphorylation. It's a good word to know. You might see it on some standardized tests or on your exams. And it's called this because you have an oxidative part. Each of these molecules get oxidized in the electron transport chain as they lose their hydrogens or as they lose their electrons. That creates a hydrogen gradient. And then that, through chemiosmosis, allows for phosphorylation. So that's another good word to know. The transfer of these hydrogens, these kind of going through this membrane selectively. This membrane, this ATP synthase wouldn't allow just any molecule to go through it. It's allowing these hydrogen protons to go through it. This process right here of this hydrogen going through it is called chemiosmosis. Chemiosmosis, another good word to know. So the entire process called oxidative phosphorylation, they don't happen at the same time. Oxidative generates the energy because the energy to push the hydrogens out, and then the phosphorylation happens as the hydrogens experience chemiosmosis and go back in and turn this little axle and then push the ATP or the ADP and the phosphate groups together. And then you can contrast that with substrate phosphorylation, since I'm in the mood to introduce you to terminology. Substrate phosphorylation. This is actually what happens when the ATP is produced directly in glycolysis and the Krebs cycle. So this is in glycolysis and the Krebs cycle. And this is where you have an enzyme directly helping to produce the ATP without any type of chemiosmosis or proton gradient. So if you imagine an enzyme, some blurb, some big protein blurb, and let's say it has the ADP. So it has the ADP there with its two phosphate groups, and then maybe it has another phosphate group that attaches at some other part of the enzyme. This enzyme facilitates without any kind of chemiosmosis or oxidation. It facilitates probably in conjunction with other energy releasing reactions that maybe are occurring at other parts of the enzyme. So maybe you can imagine a little spark right there, and then that twists this entire enzyme. This isn't exactly how it might work, but it's a good idea. And then these two things maybe get pushed together. When it's just an enzyme without any of this chemiosmosis that's driven by oxidation, like we learned in the electron transport chain, we call this substrate phosphorylation. And then the substrates are just the things that attach to the enzyme and have something performed on them. So anyway, hopefully you found this little video mildly useful.
The electron transport chain is a set of enzymes which is located on the inner mitochondrial membrane, and it is the main source of energy in the aerobic pathway of energy generation. Hello guys, and welcome to Math Simplified. In this video we will study about the overview, the concepts, the role of various enzyme complexes, and energy calculations in the electron transport chain. Before beginning, make sure to hit the subscribe button below for all the upcoming videos from Math Simplified, and also make sure to hit the like button on our Facebook page for all the flashcards and other stuff. Before watching the video on electron transport chain, also make sure to watch the videos on Krebs cycle, glycolysis, and beta oxidation. You can find the links for the videos in the description of this video. Now let's talk about the overview of the electron transport chain first. The mitochondria has an outer mitochondrial membrane, an inner mitochondrial membrane. The space between these membranes is called the intermembrane space, and inside we have the matrix of the mitochondria. If we look at a close section of the membrane structure of the mitochondria, we can see the structure of the electron transport chain. So here we have the outer mitochondrial membrane, the inner mitochondrial membrane, the intermembrane space. The inner mitochondrial membrane contains 4 sets of enzyme complexes which are named from 1 to 4 in Roman numerals. The matrix of the mitochondria also contains numerous other cycles like Krebs cycle and beta oxidation. If you have watched the video on these topics, you know that these cycles produce reducing equivalents called NADH and FADH2. These reducing equivalents contain electrons which are donated to this set of enzyme complexes. The electrons then travel from the first to the fourth enzyme complex, and in this movement energy is generated. This energy is utilized in pumping hydrogen ions into the intermembrane space from the matrix of the mitochondria. This continuous pumping of hydrogen ions into the intermembrane space causes the generation of a higher concentration of hydrogen ions in the intermembrane space as compared to the matrix of the mitochondria. This generates a positive charge in the intermembrane space and a negative charge in the matrix of the mitochondria. This is called the electrochemical gradient. The hydrogen ions cannot cross against this electrochemical gradient because the inner mitochondrial membrane is non-permeable to ions. So here we require a special transporter. The enzyme ATP synthase comes into play. The ATP synthase transports hydrogen ions into the matrix of the mitochondria and uses the energy generated from the flow of hydrogen ions to phosphorylate adenosine diphosphate into adenosine triphosphate. This is the basis of generation of energy in the form of ATP, which is a currency of energy in our body. Now let's talk about the various electron transport chain complexes. What you see here is the inner mitochondrial membrane, which has these electron transport chain complexes. These complexes are numbered from 1 to 4 in Roman numerals. And in addition to all the 4 complexes, we also have 2 additional prosthetic groups called the coenzyme Q and cytochrome C. So let's first talk about the complex number 1. The complex 1 of the electron transport chain is named as NADH dehydrogenase complex. It is also known as the NADH oxidoreductase. The first complex is a L-shaped protein complex which is present in the inner mitochondrial membrane. It has 2 arms. The vertical arm is located inside the inner mitochondrial membrane, whereas the horizontal arm is located inside the matrix of the mitochondria. It is very easy to understand the function of any enzyme of electron transport chain by just understanding its nomenclature. So the complex 1 is called NADH dehydrogenase complex because it is a dehydrogenase which removes hydrogen. It removes hydrogen from the reduced form of the nicotinamide adenine dinucleotide. And it is called a complex because it also contains flavine mononucleotides and iron sulfur compounds. It is called oxidoreductase because dehydrogenation is an example of oxidation reduction reaction. The main job of this complex is to receive electrons from NADH and transport it further to the electron transport chain. Now coming to the complex 2 of the electron transport chain. The complex 2 is called succinate dehydrogenase complex. It is called a dehydrogenase because it removes hydrogen from compounds. In this case, it will remove hydrogen from the succinate and oxidize it to fumarate, which is a step of the Krebs cycle. And similarly, it is called a complex because it also contains iron-sulfur clusters in addition to succinate dehydrogenase. The second complex is a part of Krebs cycle which catalyzes the reaction for oxidation of succinate to fumarate. In this reaction, the reducing equivalent FADH2 is produced, which is utilized for donation of electrons in the electron transport chain. The complex 3 of the electron transport chain is called the cytochrome reductase. It is also known as the Q-cytochrome C oxidoreductase. Now, cytochromes are a group of proteins which have heme as their complexes or the prosthetic groups. They also have iron core in which the iron can exist in an oxidized or reduced form depending on the electrons it has. The complex 3 contains 3 types of cytochromes: the cytochrome B, cytochrome C1 and cytochrome C. The main job of the complex 3 is to accept electrons from the electron transport chain and then transport it to the cytochrome C. The cytochrome C then transports these electrons to the complex 4 of the electron transport chain. The complex 4 is called the cytochrome C oxidase. It is a heme and copper containing complex whose main job is to oxidize cytochrome C, that is to receive electrons from cytochrome C, and then it uses those electrons for the reduction of oxygen to water, and hence it is called cytochrome C oxidase.
Welcome to our third and final video in our series on cellular respiration. In this video, we'll focus on electron transport phosphorylation. In part 1, we covered glycolysis, where glucose was broken down. We had to spend little ATP, and we ended up making 2 pyruvates. In doing so, we made 4 ATP, a profit of 2 once we spent these 2, and we made NADH, which we sent on to the electron transport chain, the topic of this video. At the end of glycolysis, we took these 2 pyruvates through the Krebs cycle. During the Krebs cycle, we prepared pyruvate in the prep step, liberating 2 carbon dioxide as waste products, making 2 more NADH that will go to electron transport. Acetyl-CoA was picked up by oxaloacetate to form citrate, and then continuing through a series of intermediates, the citric acid cycle produced 2 ATP, liberated 4 carbon dioxides, made 6 NADH and 2 FADH2, all of which are going to the electron transport chain. So if we look at an accounting of cellular respiration through the Krebs cycle, we can see that we've taken apart our glucose and we've broken it up into 6 carbon dioxides, a 6 carbon molecule, 6 carbon dioxides. We've produced only 4 of our approximately 36 ATP. We've not yet used oxygen and we've not yet used water. But we have been creating high energy electron carriers. We've been stripping away high energy electrons from the high energy bonds of glucose as we've been breaking it down. And we have these NADHs and the 2 FADH2s, which are going to go to the electron transport chain for electron transport phosphorylation, where we're going to make the majority of our ATP. So let's get to it. Here we are inside the mitochondria, and we have 10 NADHs and 2 FADH2s that we've made throughout the process. And we need to send them to the electron transport chain. And the electron transport chain is found embedded in the inner membrane of the mitochondria. So if we look in here, we have to zoom in to these folds or these cristae, and in these folds of the mitochondria, embedded in this membrane, we're going to find our electron transport chain, which I have drawn. Let's see if I can grab this and make this bigger. This may look familiar from our video series on photosynthesis. An electron transport chain This is my representation, but I'll show you another way to look at it. Here's a diagram. It's a series of enzymes embedded in a membrane, and electrons get passed along, and in doing so, we're going to make ATP. So let's look at this in isolation. This electron transport chain is embedded in the inner membrane of the mitochondria, and as electrons get passed along, energy is given off to make ATP. So, for instance, NADH, a high energy electron carrier, comes to the electron transport chain, drops off that high energy electron in the hydrogen, and as the electron goes down in a series of reduction-oxidation reactions, energy is given off to make ATP. Kind of. I say kind of because it's not that the energy is directly given off to ATP, but this process is going to set up the conditions under which ATP is made. So we need to look at the system in a little different way. So here's the inner membrane of the mitochondria, dividing us into an outer compartment and an inner compartment. So we might have, let's see, on this diagram, here's the inner compartment, and here is the outer compartment. So it's across this membrane that the action is going to occur. So here's that inner mitochondrial membrane, and here's the electron transport chain. The 10 NADHs and 2 FADH2s made in earlier stages of respiration come to the inner compartment and deliver those high energy electrons to this electron transport chain. Now, whatever happens to NADH, the same thing is going to happen for FADH2, so we're just going to look at NADH just to simplify things. As this high energy electron gets loaded onto this first step of the electron transport, it has to bring this hydrogen with it. It needs to bring this proton. It can't go into this molecule alone. So as it travels through this first protein complex, the electron carries the proton with it, the H+, and essentially pumps it into the inner compartment, or from the inner compartment to the outer compartment. And then the electron gets picked up by an electron acceptor. That electron acceptor brings the electron over to the next protein complex. As the electron drops off onto this enzyme, it has to pick up another proton. So it grabs another proton from the inner compartment and pulls it along with it through this protein complex. And once again, we pump a proton from the inner compartment to the outer compartment. The electron gets picked up by another electron acceptor and gets delivered to the final enzyme or protein complex in this membrane. For a third time, as this electron is loaded onto this protein complex, it has to grab a proton from the inner compartment, and it pumps this proton across. So essentially, this electron transport chain is a three-part proton pump, and this electron is no longer a high-energy electron. Now, imagine this happens over and over again with all the NADH delivering their electrons and all the FADH2s. We're going to pump a lot of protons into the outer compartment. Let's move all those to the outside. So we have all these protons out here, and these protons are trapped in the outer compartment, and they want to move down their concentration gradient back into the inner compartment, but they can't make it through this membrane. Well, it turns out there's a special channel for them, and the channel is called ATP synthase. It will allow the protons to flow back through down their concentration gradient. And notice it's called ATP synthase. It is also not only a channel protein, but it's also an enzyme that catalyzes the ADP plus P reaction to make ATP. However, if you recall, this reaction also requires the input of energy. The energy for this reaction comes from the flow of protons through this channel. And this is electron transport phosphorylation. We phosphorylate ADP into ATP, and it's driven by chemiosmosis, which is the production of an electrochemical gradient that then drives the production of ATP. And it's responsible for the final 32 ATP that we're going to make. So let's quickly summarize. In electron transport phosphorylation, electron carriers take the high energy electrons picked up during glycolysis, the prep step, and Krebs cycle, the NADH and FADH2, to a group of enzymes in the folds of the mitochondria. These enzymes take the high energy electrons and pass them down the line, and as the electron is passed, hydrogen ions, or protons, are pumped to the outer compartment of the mitochondria. This ion gradient is used to run the ATP production by electron transport phosphorylation, or chemiosmosis, and we produce approximately 32 ATP. So let's move to a final accounting. After we go through the ETC, and we've dropped off our higher G electrons from the 10NADH and the 2FADH2, we have an output of approximately 32 ATP. So, we're done making ATP. But we have a problem. We've yet to make water, and we've yet to use oxygen. So we need to go back to the picture. When the electron gets to the end of the electron transport chain, it's got to be picked up and moved out of the way so that the next electron can come down. It's got to move from the bottom of the staircase so the next person can come down the stairs. So something's got to remove this electron. That something is oxygen. We finally use oxygen. This is aerobic respiration, cellular respiration. Oxygen comes in, grabs the electron, and grabs a hydrogen to make water. And so there we have it. We finally use oxygen, and we finally make water. So we're done. When the electron gets to the end of the electronic transport chain, oxygen is the last electron acceptor, and water is the last product made, as the oxygen picks up the electron and combines with a hydrogen. This is interesting because it's a nice mirror to what we saw in photosynthesis, where water was the original electron donor. Here, oxygen is the last electron acceptor, and water is the final product made. So now we're finished in our three-part series on respiration: glycolysis, the Krebs cycle, and electron transport.
The F1F0 ATP synthase plays a critical role in the synthesis of ATP, which, as we all know, is crucial for most of life's processes. In this lesson, we're going to focus on the F1F0 ATP synthase, which is one of my favorite enzymes, and I think a favorite of many people that love enzymes and that love protein structure. We will explore what is called the binding change model that Paul Boyer first proposed for how ATP synthesis is achieved, and then apply a variety of experimental approaches to confirm specific aspects of the synthase's mechanism of action. The structure and functional mechanisms of the synthase were discovered by the combined work of Paul Boyer, a biochemist at UCLA, and John Walker, a structural biologist at the MRC in Cambridge, and represents one of the most important collaborations between biochemistry and structural biology in recent memory. For their efforts in uncovering the structure and function of this enzyme, Boyer and Walker shared the 1997 Nobel Prize in Chemistry with Jen School. At a mechanistic level, we will explore how the F1F0 ATP synthase harnesses the energy of the proton gradient in mitochondria to synthesize ATP. This will help us really explain how the protons pumped by the electron transport chain actually provide the energy for ATP synthesis. To understand the workings of this truly amazing molecular machine, we will define the major protein components of the synthase complex, and then what they contribute to its mechanism of action. The F1F0 ATP synthase is an example of an F-class pump. There are other classes of transmembrane pumps that hydrolyze ATP to actively transport cargo across membranes. But in the case of the ATP synthase, it utilizes the movement of protons across the inner mitochondrial membrane to drive the synthesis of ATP. The ATP synthase has multiple protein subunits that are clustered into two major components: the so-called F0 complex and the F1 complex. The F0 complex is a proton channel and is where protons flow across the inner membrane of the mitochondrion. The F0 contains three different subunits, A, B, and C. The C subunits assemble into a ring-like structure within the inner mitochondrial membrane, and the proton channel lies at the interface between this so-called C ring and the A subunit. In contrast, the F1 complex is the site of ATP synthesis. It is composed of 5 types of subunits: alpha, beta, gamma, delta, and epsilon. the components of the synthase most directly involved in the synthesis reaction are the three pairs of alpha and beta heterodimers. Together, these three pairs form a hexamer and comprise the mushroom head of the synthase, which faces into the mitochondrial matrix. This is the site of ATP formation, where actual enzyme catalysis is performed by the beta subunit. The gamma subunit links the hexamer to the inner portion of the C ring of the F0 complex. It is the rotating axis, therefore, of the complex of the synthase. The delta subunit, on the other hand, anchors the hexamer to the B subunit that is embedded in the membrane. The delta subunit effectively stabilizes the hexamer and holds it in place, and prevents the hexamer itself from rotating. Thus, when the C ring rotates, it in turn rotates the gamma and epsilon subunits, but does not rotate the hexamer itself. We are looking at the turning gamma subunit and the epsilon subunits, and there's the C ring in the membrane. The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane. There's the stalk, including the delta and the B subunit. The alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We will now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic phosphate diffuse into the active site, and the ATP has just been released, and simultaneously the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its two phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. Synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets and restarts the synthesis of another ATP molecule. This is a summary of the events at the active site. It is also intriguing just how the C ring rotates. We can start with the analogy of a water wheel. We can easily grasp the idea of how a water wheel rotates. It's driven by the flow of water, allowing the wooden fins to go in a particular direction. Thus, the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite direction. Here we see the C ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space. And there's a separate egress channel for protons on the matrix side. In this animation, you see protons flowing out from an interface between the C ring and the A subunit. But what drives the specific direction of this rotation? For example, if we were looking down, it is always a counterclockwise rotation. Why and what drives that? On each of the C subunits within the C ring, there is a proton binding site. Notice the high concentration of protons on the intermembrane side and their flow to the lower concentration on the matrix side. Protons load from the highly concentrated intermembrane side onto the C subunits, and they are released on the matrix side where the concentration of the protons is low. But why can't protons go around the other way as well? In principle, just as in the water wheel analogy, the C ring should be able to rotate in either direction, shouldn't it? There are two interconnected reasons for this unidirectional movement. The amino acid side chains in C subunits would repel the bound proton, so that when a proton loads from the intermembrane side, the C ring cannot rotate back in the opposite direction because it is energetically unfavorable. Rotating counterclockwise, on the other hand, is energetically favorable because there are no repulsive interactions. Remember, there's a concentration gradient that favors binding of protons on the intermembrane side and the release of protons on the matrix side. So in summary, the proton gradient determines the direction of the rotation, favoring movement in the counterclockwise direction of the C-ring. This avoids unfavorable interactions with the amino acid side chains of the C-ring. The rotating C-ring is composed of 8 to 12 copies of the C subunit. The protons pass through a channel that is formed at the interface of the C-ring and the A subunit of the F0 complex. When the C-ring rotates, the gamma and epsilon subunits rotate along with it. However, as I mentioned before, the alpha-beta hexamer of F1 does not rotate because it is anchored by the delta subunit. The delta subunit is in turn bound to the B subunit, which is embedded in the inner mitochondrial membrane. Let's situate ourselves within the mitochondrial matrix so that we can look down on the hexamer. In this view, the gamma subunit is in the middle, surrounded by the 3 alpha-beta heterodimers. We will in particular focus on the beta subunits, where the actual catalysis takes place. In Boyer's explanation of ATP synthesis, which is also known as his binding change mechanism, each beta subunit can exist in one of 3 different conformations at a given time. One of the conformations is called open. In the open conformation, the substrates of the ATP synthesis reaction, namely ADP and inorganic phosphate, can bind to the open beta subunit. Another conformation is called loose. Although it seems a little bit counterintuitive, in the loose conformation, both of the substrates are associated with the beta subunit. They could be considered to be loosely locked into the active site in comparison to what is the next conformation. This third and final conformation is called tight, and this is where the substrates are pushed together in an optimal geometry to catalyze the synthesis of ATP. All 3 different possible conformations of the beta subunit, open, loose, and tight, exist at the same time among the 3 different beta subunits in the F1 hexamer. Boyer's proposal of a binding change mechanism suggests that as the gamma subunit rotates, each beta subunit sequentially moves through the open to loose to tight conformations. Let's start with beta 1 in the open configuration, beta 2 in the loose configuration, and beta 3 in the tight configuration. As the gamma start rotates 120 degrees, beta 1 switches from open to loose, locking in the substrates. Beta 2 in turn goes from loose to tight, driving the catalysis of ATP. And beta 3, that was originally tight, switches to open, releasing the ATP product. As the gamma subunit continues to rotate, the sequence continues. Beta 1 now goes from loose to tight, driving the catalysis of ATP. Beta 2 goes from tight to open, releasing the ATP product. And then beta 3 goes to loose, locking in the substrates that came in after the original ATP product left. Rotation of the gamma subunit causes the protein conformations to change within the beta subunits, varying each subunit's binding affinity for the substrates. The catalytic effect of the beta subunits will also be different, ultimately leading to the release of the product. Once ATP is released, new substrates can then come in and bind. And as the rotation continues, the cycle of ATP synthesis repeats. It is worth noting that the beta subunits cannot all assume the same states at the same time. 3 beta subunits are therefore staggered in conformation relative to each other. So there's a domino effect of structural changes that propagates around the 3 beta subunits of the hexamer. The F1-F0 complex converts the mechanical energy from the proton flow into the rotation of the gamma stalk, which is then transformed into the conformational changes in the beta subunits of the hexamer, that in turn is converted to the chemical energy in the bonds of newly synthesized ATP.
All living organisms on Earth are made of cells, and all cells are using ATP as an energy coin. Although ATP is a tiny molecule, it can yield considerable amounts of energy through the simple process of reacting with water. They resemble rechargeable batteries. Those in a state of charging are called ATP, and those in a state of discharging are called ADP. ADP, a discharge molecule, becomes ATP, a charged molecule again, by using the energy of food. The mitochondria in cells contain a factory powered by food that generates ATP molecules. By extracting energy from the ATP produced at the factory, cells carry out activities of life, such as movement, growth, and protein production. We carry batteries around that can easily be used when required. In a similar way, cells extract energy from ATP molecules according to their needs. The factory in mitochondria generating ATP is actually an enzyme called ATP synthase. It plays an extremely critical role because it is responsible for generating the energy required to maintain life. Since the 1990s, the mechanism and action of ATP synthase have been elucidated by a group led by Professor Yoshida of Kyoto Sangyo University. During cellular respiration, a concentration of hydrogen ions creates a potential difference between the inner and outer mitochondrial membranes. ATP synthase has two rotating portions, called FO and F1. The FO portion rotates using the force of hydrogen ions flowing from the higher concentration level to the lower. The rotation energy generates ATP in the F1 portion. The mechanism of ATP generation is very similar to that used to produce hydroelectricity. Hydroelectric power generation uses the gravitational force of falling or flowing water to generate electricity. In the case of ATP synthase, a difference in the concentration of hydrogen ions is used to generate ATP. The size of ATP synthase is 10 nanometers, or 1/100,000th of a millimeter. It's been discovered that each cell contains numerous rotating nano-sized factories producing energy. Many mysteries concerning ATP synthase remain to be solved, including the mechanisms that regulate ATP synthesis. ATP synthase synthesizes ATP when there is a difference in the concentration of hydrogen ions. But it consumes ATP when the difference disappears as the result of a reverse reaction. When the difference in the concentration of hydrogen ions cannot be created for some reason, such as a state of starvation, some kind of mechanism should work to prevent the wasteful consumption of ATP that has been synthesized with so much effort under critical conditions. In cooperation with Professor Gregory Cook at the University of Otago in New Zealand, the ATP Synthesis Regulation Project at the Japan Science and Technology Agency has unraveled the regulatory mechanisms of ATP synthesis by carrying out enzymatic reactions one after another. The regulatory mechanism of the human ATP synthase is also gradually being clarified. The project team attached a bead to the rotating part of the F1 of the human ATP synthase and successfully observed its rotation. Here are the images obtained. They reveal that the human ATP synthase rotates at an incredible speed, around 1,400 times per second. These are slow motion images, 1,000 times slower than the actual speed. In the case of humans, a protein factor binds to the motor from the outside to inhibit rotation. These test images show that rotation stops when an inhibitor protein is added. It's believed that this mechanism controls the useless consumption of ATP. The project has also elucidated the ATP synthesis regulation mechanism of various other living organisms, including plants. It's hoped that in the future, the research results will provide new information leading to the development of breakthrough therapy for diseases such as metabolic energy disorders.